Hello, everyone. Welcome to Moberg Analytics Case Studies in Neurocritical Care AI. I'm Craig Maddox, CTO of Moberg Analytics. I'm joined by our founder and CEO, Dick Moberg, and our VP of R&D, Ethan Moyer. Welcome to what I believe is the 20th of these talks. As you know, brain injury is a big mission. These talks and a lot of other things we do are our part in helping advance that mission. We have these webinar series with case studies and examples. We have low cost and starter grants to help people get started if they want to do neuro-related AI work. We can offer consulting tools and software platforms. Another exciting extension to these is next month at the Neurocritical Care annual meeting in Montreal. We're going to be co-leading a workshop, kind of an extension to these webinars. We're going to be going over the topics of patient metadata and harmonization, data formats, converting, data cleaning, regulatory compliance, and then we're going to go through a couple of examples with cerebral autoregulation and some visualization and modeling. And some of you on this call are going to be co-presenting with us, so we're really excited about that. Again, that's just kind of an extension of getting AI into our field. Please, if you have any feedback, you can email me directly or provide feedback using the QR code or the bit.ly link there for feedback. We would love if more people step forward and volunteer to share the work they're doing. We come across really cool work being done every day, and so we would love to get others up here on our webinar and share it because it helps everyone in the community. And it's really cool to see what everybody's doing. It's amazing. Every week we're just blown away by the amazing work that people are doing. Don't forget that the replays are out there. I've almost exhausted the space on this slide, maybe another week or two, and we're not going to be able to read it. So anyway, the replays are all out there on our YouTube channel. And if you have questions, please use today the Q&A feature that we all know and love so well. So today we are super excited to welcome Dr. Mypinder Sekhon. He's the division head and clinical associate professor at the critical care medicine at the University of British Columbia. He's also an intensive care physician at Vancouver General Hospital. He graduated there from UBC, focusing on internal and critical care medicine. He then did a neurocritical care fellowship at Addenbrooke Hospital at University of Cambridge in England. His research is focused on the biological mechanisms related to cerebral ischemia. And today he's going to discuss his work in the characterization of the physiology during circulatory arrest. I think Dick has known Myp for a long time. I met him last year. I think Ethan and I met him last year, and he kind of came back into our radar because it turns out he treated a friend of ours, Jamie Mocrazy, back in 2015. She was an Olympic-bound freestyle skier who had a horrific accident on a half pipe. Paramedics had all but written her off, but she had the good fortune to be brought into Myp's care at Vancouver General. To this day, she credits him with saving her life and considers him family. I think he even attended her wedding not long during her healing process. So with that, Myp, I will stop sharing and turn it over to you and take it away.
Great. Well, thanks so much, you guys. Really appreciate the opportunity to present to you about and the very kind introduction. The work I'm going to present is fairly exploratory, but serves, we think, predominantly as a proof of concept and as a model of studying cerebral ischemia in vivo within the human body. And where I think there could be tremendous collaboration or insights from your expertise is harmonizing some of our data collection to really leverage the power of that. So with that context, I'll talk to you about what we're doing now here in Vancouver, which is studying the physiology of the human dying process as a model of circulatory arrest of brain ischemia. I have no conflicts for this work whatsoever. We're all neuro-inclined people here, so I don't have to convince you that this is the most important organ in the body. dare I say, the least understood and the most complex. And it's the connection between the heart and brain, the two most vital of all the vital organs, that governs what we know to be life and serves as the boundary of between life and death. And perhaps the most important mechanism in the body, pathophysiologic mechanism, is when the most important organ is lacking the most important molecule, that being oxygen, and cerebral ischemia. To highlight this, I'm going to tell you about a case. I have permission to share this gentleman's case. His name was Michael, 26 at the time. In February 2023, he was admitted to our ICU after cardiac arrest. And on day 4, his MRI revealed these dreaded words of really widespread hypoxic ischemic brain injury throughout the cortex, throughout key areas of the thalami, the hippocampi, the basal ganglia. And his parents, David and Laura, following this, plus his clinical exam and after prognostication, made the very difficult decision to proceed with palliative care. Now, it was their wish that he be considered for organ donation. And so we tried to operationalize that. Now, he was not neurologically brain dead. So how we do that here, much like I'm sure in many of your jurisdictions, is we remove the endotracheal tube, provide the patient with palliative care as we normally would. And if the patient passes within a defined period of time, they may be considered to be an organ donor. So that process being DCD, or donation after cardiac death. Now, Michael kept breathing and his heart kept beating beyond our 2-hour limit. And so when we went to go inform his parents that it wasn't going to work out, his father said something. And his father said, well, we've looked into his eyes, he's gone. And what was he getting at? Well, what is the eye? We think of our eye, medical textbooks have written about papilledema, pupillar light reflexes. It's our only physical manifestation or window into the human brain. And of course, that occurs via the optic nerve, through the optic chiasm, Meyer's loops, and then into the occipital cortex. So it's a pathway that traverses not only the midbrain, the extracranial nervous system, but also the cerebrum as well. And we think what maybe Michael's father, David, was getting at was his view of what death meant. And in Canada, we have a unified definition of death, which is defined off the permanent cessation of brain function. And that can occur at the result of cessation, or the moment of cessation of blood circulation to the brain after circulatory arrest. Clinically, though, as it pertains to DCD, what we do is we define the moment of death to be the time at which the arterial pulse pressure is less than 5 with a radioarterial line. So here's a discordance. We have a brain-based definition of death in Canada. So our conceptual definition is brain-based. However, clinically, we're using a metric of what is in the radial artery. Alexander Monroe told us 250 years ago that the quantity of blood that circulates in the brain is greater than most organs of the same weight. Thus, one of our arms will be found to weigh more than our brain and cerebellum, but the area of the two vertebrals and carotids joined together is much greater than the subclavian. This was the first documentation in the medical literature that suggested that the brain circulation and physiology was different than that of the extracranial tissues. And that was written in this thesis in 1783. Now, contemporary visionaries have actually also made this observation. If you scour Taylor Swift's lyrics, at one point she says, "I can feel a pulse, but I got nothing in my brain." That's what people say. That's what people say. Hoo. So not only is Alexander Monroe made this observation, but in our lay media, Taylor Swift has as well. And so did Michael's father, David. And our critical assumption right now is what is happening in the brain is simultaneously happening in the radial artery. Our issue is we have diverging opinions. Clinically, we equate the radial artery to be what is in the brain. However, functionally, they may be different. That is a hypothesis. That is our brain the same? Is our brain's physiology, our circulatory physiology in the brain the same as what is in the extracranial circulation? Well, when we look at what David said, remember he said we looked into his eyes, he is gone. My colleague and I rushed back into the room at that time and we took a picture of the monitor. And the monitor at which point his father said, or after he said that, his blood pressure was 75/36. And so here we have this conundrum. We have a father who has ascertained that his son's brain is absent of flow and function, whereas his son's extracranial circulation is still flowing. So how can we study this process and reconcile this issue? Accessing the most difficult organ to access in the human body, um, but studying it in an ethical manner. Well, if we think of what the dying process is, it's ubiquitous, it happens to everyone. But it's also thinking about it from a physiologic perspective, it's a stimulus. And that stimulus is characterized predominantly by cerebral ischemia. Um, and it's an opportunity to study the response. So it could be an opportunity to study the response of cerebral ischemia in the most important organ in the body, lacking the most important molecule that sustains its function, but also one that could help inform insights into resuscitation sciences, stroke medicine, and potentially organ donation practices. So that led us to this study to characterize the cerebrovascular physiology of circulatory arrest in humans. Initially, when we set on the path, we had 4 aims, and we wanted to do this in a comprehensive manner. The first was, can we even study this process in humans in a prospective and multimodal manner? If we can we study the heart and the brain at the same time in terms of as it relates to the timing and relationships of circulatory arrest and relationships between the physiology of both organ systems? Third, can we take advantage of this opportunity to identify biomarkers that could reflect cerebral ischemia? And finally, could we correlate postmortem pathology to the observations, the physiologic observations made to explain what we're seeing? This is we're fortunate to have this manuscript will be out in the next month or so. And this was the initial sort of experimental setup. We decided to go with a non-invasive approach to measure brain physiology with an automated transcranial Doppler of the MCA and PCA, near-infrared spectroscopy, and then uniquely positioning of an oximetric jugular venous bulb catheter sitting just distal to the sigmoid sinus. For the heart, though, of course, we have all the standard telemetry and radial artery, but we also place, as part of this work, a premorbid, at the time of palliation, a Swan-Ganz catheter to characterize cardiac output and oxygen saturation within the pulmonary artery. Patients are then cared for as per normal for their palliative care. But uniquely, we also collect serum specimens and plasma specimens before palliation and then immediately after the patient passes to identify any sort of biomarkers or changes in the proteome or metabolome that the brain is exhibiting. Now, we collect these specimens from the radial artery catheter, but also simultaneously from the jugular venous bulb catheter. And the reason for that is any one moment in time, the differences in concentration at the arterial vasculature and the jugular venous bulb would reflect transcerebral gradients of biomarkers. And finally, the patients in this particular study undergo a brain autopsy and heart autopsy. Now, ethically, I don't care for any of these patients. that would be, of course, a huge conflict of interest. So these are patients who are referred to on the decision has already been made for palliative care, and are cared for by our colleagues. I don't have to tell you, obviously, what a Swan-Ganz catheter looks like, but there's a positioning in the neck, and the catheter is positioned at the pulmonary artery right there, proximal pulmonary artery. And of course, patients have a radial arterial catheter. And so that measures mechanical and electrical heart function. For the brain, we have, as I said, automated transcranial Doppler, which has really kind of been game changing for this work because we don't have to be in the room. We are in the room to observe the dying process, even with the families there, but we're not having to hold the transcranial Doppler for continuous insonation. This particular device from Neurosignal will continue, even if the patient does cough or move, continually reacquire the window to make for, it's not perfect, but make for relatively continuous data collection. The jugular venous bulb is inserted most of the time into the right jugular vein, so that's going cephalic, and then the Swan-Ganz catheter is, of course, going down. So this allows not blood flow, but blood flow velocity, and a measure of oxygen exchange during the dying process. This is what the bedside looks like. It's a lot of stuff. And you might say, well, who the heck would ever consent to this sort of study? And this is where the feasibility of the data becomes very important. This is, for me, perhaps one of the most important findings. That in the first 32 patients, on the y-axis, we have individual numbers plus their underlying disease states. And on the top there, we have the various different data points that were collected across each case. PAC, meaning pulmonary artery catheter. Orange means collected. Blue means we didn't manage to get that in that particular patient. And there's a lot of orange on the page. Now you might say, okay, well, in the first 10, there's no pulmonary artery catheters. That's because we didn't have consent at that time, and so this was an amendment. But since that point, the vast majority do. Currently, we're at approximately mid-40s right now in our current cohort. And one thing to notice, there's a lot of orange in the column with biomarkers, and there's a lot of orange in the column with brain autopsy. This is perhaps the most popular part. Perhaps popular is not the correct word to use for this particular study for families, but this is by and large, the reason why a lot of families opt in is because they get an autopsy as part of this work, and that report gets provided to patients. And sometimes we see, not infrequently, things on the autopsy that not only help characterize the physiology, but are also really important for the care and screening for future family members. So for me, this was aim one. This is a proof of concept that can we collect this data? Can we enroll patients? Can we consent patients? And the answer was yes. Our consent rate for all comers for this work is just slightly in excess of 70%. The second thing we wanted to look at was timing. Is there a difference in timing of circulatory arrest of the body versus the brain? Here, this is just T0 as a time of extubation. And this just highlights that per disease, stratified by disease, there's no real pattern. Whether it's hypoxic ischemic brain injury or patients with sepsis or TBI, the data is kind of all over the place. And that might not seem super surprising, but I think it's important to show heterogeneity across patients. And we know that from clinically, that after palliation, of course, different patients will pass it at different timelines. One thing that was really interesting, though, was now if we take T0 to be the time at which brain blood flow velocity ceased on the transcranial Doppler. And now, make no mistake, I firmly believe TCD is obviously not a gold standard. It is a surrogate measure. Having said that, in each case, in dark blue is the time from absence of TCD flow velocities to then absence of systemic circulatory flow as defined by a pulse pressure of less than 5. From the light blue is really interesting. That's the time from PEA to then the time of electrical asystole. So I firmly do not think, even in Canada, that this changes our definition. It does not change our definition of death. However, it does highlight that perhaps in some patients, the brain's circulatory physiology may cease prior to that of mechanical and definitely electrical heart function. A bit more provocative. Now, something that we're doing, and this is something that I'd be super keen to collaborate with Dick and Craig and Ethan on, is we're studying this in a more comprehensive manner. So in our initial cohort of patients, we did this with non-invasive monitoring. Now we're actually doing this with parenchymal monitoring and bihemispheric parenchymal monitoring. So we place a quadruple lumen bolt with a Bowman perfusion catheter, Lycox catheter on both sides, and EEG depth electrodes. And on one side, a microdialysis catheter, and on the other side, the ICP catheter. This is what the monitoring looks like prior to palliation. It's a lot of stuff. We also have somatosensory evoked potentials, brainstem auditory evoked potentials, and surface EEG as well. We do a CT scan before to just document the placement of the catheters within the parenchyma, and then conduct a perfusion scan to characterize the symmetry of the perfusion bilaterally. Most of these patients will have an MRI. In this particular case, this patient had a cardiac arrest and diffuse restricted diffusion of the cortex. So that is without a question a limitation of this work, is that these patients are entirely, almost all of them, brain injured. So firmly, I will also acknowledge we're not necessarily describing normal brain physiology. I think it'd be almost impossible to do so ethically, but at least we're able to do this in humans and provide some insights. This is what the sort of more comprehensive neuromonitoring looks like. We conduct these cases in the operating room. That's all the monitoring. There's on the left-hand side of the screen, the EEG and somatosensory evoked potentials, and then all the rest of the parenchymal monitoring here on the bottom. We've got the transcranial Doppler as well, and then the bedside monitoring for Swan-Ganz and all that. As I said, this is kind of what the monitoring looks like, the bihemispheric parenchymal monitoring prior to palliation. Microdialysis pump is at the bottom here. And what do we see? Well, in this particular case, we've done 3 of these cases so far since the spring. One thing that was really interesting is we see similar patterns as to what we see with our TCD. So mean arterial pressure on the top, intracranial pressure below that, and then pulse pressure. So systolic in green, in pink is the diastolic, and the right and left PbTO2. And in fact, the right and left PbTO2 gave out approximately about 10 minutes before PEA was reached. Importantly, the TCD, in this case, it was the anterior cerebral artery flow velocity, gave out just at around the same time as when the PbTO2 gave out. The microdialysis data was super interesting, and we've seen a similar pattern in 3 cases so far. In that glucose before WLSM is withdrawal life-sustaining therapy. So the first readings here are patients prior to palliation, and then we get a sample during the dying process, and then post-mortem. And we see a rise in glutamate and LP ratio, but predominantly glutamate after death has occurred, which the etiology of which is certainly something to debate and discuss. This is a picture of the waveforms. Now we collect waveforms in this newest cohort of patients. We have the TCD waveform. The one downside of the automated TCDs is the stochastic movement that you get when the device is trying to search for a signal. So when it loses a signal, it starts going zzz, 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 and so that will create numerical values. So we have to clean the data for that. And that's probably responsible for this rise in numerical ACA flow velocity. I don't think that's actually physiologic. That's all this stochastic movement. And then the EEG data provides a really nice electric correlate to go along with the cerebrovascular correlations. Just getting to the rest of the aims here. So that was all feasibility. Yes, this is feasible to study this kind of work in a comprehensive manner. Are there things that we can look at in terms of relationships? This is just very preliminary. One thing that we do see is, by and large, when we plot MAP against the flow velocities, the relationships are linear. And now, does this mean that the Lasson's curve is not real? Well, I mean, I'm skeptical of Lasson's curve, period. However, these are severely brain injured patients. So the loss of the myogenic mechanism and the linear relationship is likely just related to that. However, there's a lot of within patient heterogeneity. So each gray line is an individual patient, and then the black line is the summation across the entire cohort. So that is important in and of itself. It's a lot of between patient physiologic heterogeneity. The Swan-Ganz catheters and jugular venous bulb. Well, why do we do that? This is really to compare the oxygen uptake of the brain versus the extracranial tissues during the dying process. And one thing that we do see across the cohort is, again, as mean arterial pressure starts to give out, the oxygen extraction fraction does seem to rise for the extracranial tissues as opposed to the brain. Again, my suspicion for this is twofold. One, these are severely injured patients. So the brain in and of itself is going to be out of proportion much more injured to the extracranial tissues. And two, we do provide not a lot of sedative, but some sedative use during palliative care with narcotics and benzodiazepines to ease any sort of signs of pain and or suffering. So I suspect that the O2EF is definitely confounded by those two issues. One other thing that's kind of interesting that we've managed to capture is the physiology of agonal breathing. And the down deflections there on the waveforms of the pulmonary artery catheter pressure are when the patient is taking some agonal breaths. And what we do see are some brief resumptions of PCA blood flow velocity in those patients. The theory here being that with every agonal breath, central venous pressure rises due to and causes an increase of brief increase of cardiac filling pressure, which then translates to a brief increase of mean arterial pressure, and then a brief change of some PCA blood flow velocity. Now, PCA blood flow velocity is 0 here and has gone up to 50. I want to be clear that that does not equate a lot of blood flow. I suspect that this, again, numerical change in PCA blood flow velocity is a little bit of blood flow through a very constricted vessel. But what we do see in terms of agonal breathing, in terms of the time from gas, the dotted plumb lines here are the time from the last gas the patient has taken. And then individual blue lines are the change in the physiology in the seconds following that, in hemodynamic variables on the top, in brain variables on the bottom, in oxygen variables on the, or brain variables, I should say, in the middle, and the oxygen variables on the bottom. And we do see a kind of again, data is very heterogeneous, but kind of a change in PCA blood flow velocity across patients, but not a lot of changes in the other variables. Now, that's not to say that in a non-critically ill patient, oxygenation would not improve. It probably would. These are also patients who've had pretty solid lung injury as well. So I think that probably blunts the change in SpO2 that we didn't see. Kind of interesting. As I said, this work has a lot of implications for what we think is resuscitation sciences. There's been in the literature this move for head-up CPR, which I've always been very skeptical of. And interestingly, here I'll show you a couple of videos. Here's one where we've got MCA and ACA blood flow velocity. I put the head of the bed at 30 degrees from supine. The velocities went away. And then we put the bed back down at 0 right now. And the velocities came back. This was at a blood pressure approximately 70 on 50, not that dissimilar from the low flow states that CPR would provide. It wasn't just this patient though. Here's a second patient. Same thing. So on the right there is probably distal MCA, M3 maybe, M2. Same thing. I put the head of the bed at 30 degrees. Blood flow velocities disappeared at 30 instead of supine. And then went back to supine, and there's blood flow velocities coming back. So this kind of work is also has a ton of importance for resuscitation sciences as well, to the ability to study extreme low flow states. I'm just going to finish up here now. You might ask, well, why do we get all these biomarkers samples? Well, we wanted to try to identify perhaps maybe a brain troponin, something that would indicate in the moment in vivo cerebral ischemia. Thus far, our standard biomarkers that we measure, GFAP, NFL, tau, UCHL1, they didn't show any differences. We have, that is despite the staining on pathology demonstrating GFAP, NFL, tau, and a neuron cell body for UCHL1. So there is injury in the brain, it just doesn't get reflected during the ischemic period. And I suspect it's just not enough time for the proteome to change and to release things into the circulation. Now, that could be very different with a high amount of reperfusion, which obviously we can't replicate in this model. This is the biomarker, lack of biomarker change is also not related to the quantifying the amount of ischemia a patient had and or hypoxemia. We have conducted some unbiased proteomic analysis for hundreds of different novel unknown proteins. Same thing, the proteome doesn't really change. We are in the fall now conducting some metabolomics though, because the metabolome obviously is much more dynamic. So that might be, there might be some information in there. Finally, autopsies. This is, my dad's a pathologist, and he always said to me, the truth is always in the tissue. And so this was part of the reason to try to accrue autopsies in the patients to characterize our physiologic findings. And this has actually been quite invaluable, and also serves as, and we biobank these tissues, so it serves as an opportunity to use them for many, many more years to come. The vast majority of patients, patient families consent to the brain autopsy and hearts. And we also collect spinal cord autopsy samples as a neural tissue control, as an extracranial neural tissue control. We haven't done anything with that yet. And sometimes we see things that are really informative. So for example, on the B panel, this is an MCA clot directly in M1, explains why in this particular patient, we lost the MCA flow velocity so far in advance. On the A panel, this is a patient didn't really have good PCA blood flow velocities, and this was a large hemorrhage secondary to a tumor within the brainstem. And sometimes we also see things that are unexpected. This was a young gentleman in his 30s who was diagnosed with schizophrenia. He had two daughters, 4 and 8, and fortunately was kind of lost to his family due to his mental health struggles. Yeah, he was a hockey player growing up, and in fact, his brain autopsy came back with all this tau. This doesn't show all of it in detail, but he had a tauopathy, and it turned out he had multiple concussions, and so he actually had CTE. So that was important, obviously, for his family to know, but also meant that the life insurance company paid out um, his policy and is now in a trust for his kids. So this is a research. This is I suppose a result of the research that can't be encapsulated as part of a p-value or an odds ratio, but has very real world and real life significance for patients' loved ones. Finally, just to end off, going back to the case in Michael's case, his parents, in the end, I suppose they were right. That when his father had ascertained that perhaps his son had died at that blood pressure, perhaps he was right. Perhaps his son's brain function was absent, even while the extracranial circulation was going. The picture I showed of the eye before is an actual picture of Michael's eye. And his mom sent it to me for these presentations. And she said something very poignant in the email, and that was that we saw the world through his eyes, and that's how we knew he was gone. On the bottom right of the screen is Laura and David, Michael's parents, and this is them with our research team at a conference in Toronto where we presented this work last year. They're patient partners as part of this work, and so we meet with them regularly to update them on the research, and also they've in fact advised one of the aims, and that was the pathology aim, to provide some answers for patients and their loved ones, and for that valuable contribution to science. A lot of people to thank, and a lot of societies to thank for this work, and I'm not going to go through everybody. Apart from, I'm going to highlight a couple people. Jordan Bird is the PhD student who's done all this. He's just he's not a PhD student. This guy's you know he's a special talent. He's really looking forward to all the great things that he does in his career. But he's the one who's kind of pulled all this together. So I'm very humbly here on his behalf. And then a lot of our colleagues and collaborators who have made this work possible, and especially Laura and David, who are Michael's parents. And with that, I'll stop and happy to take any questions.
Thanks very much, Mip. This is amazing work. Just to remind our attendees, if you want, can you see the Q&A, Mip? You should be able to. There's one in there now.
Oh, yeah, I can see it.
Okay.
Yeah, thank you for the question for NIRS. Yeah, I didn't show our NIRS data. Unfortunately, it was there was really no relationship with hypotension. Now, with the caveat that we're using quite an old device, it's the INVOS device. It's probably like 20 years old. So I mean, I know there's advances in the technology. So I didn't want to kind of throw NIRS under the bus. So I didn't show those data. But it was essentially a flat relationship. There was no relationship. So I say that with the caveat that I don't think NIRS is not useful. I think it is useful. It's just in our particular study, we didn't see a relationship. Jeanette said many things. I'm particularly interested in the depth EEG. How many electrodes do you use? How did you assess the signal quality in this patient population? Did you observe any interesting patterns? Yeah. So we used 2 electrodes. They're 8 lead. In fact, I'll show you. One sec. Yeah. Okay. So, oops. Here we go. Okay. So here's the the depth electrodes, I think, hope you can see my um, my mouse, but it's like the little white um, sort of cap probe on the end there on the longest lumen. There's one on both sides. So those are the depth electrodes that we use. They're from Deptathlon, I think. They're 8 lead depth electrodes. To be totally honest with you, we haven't even scratched the surface of what the patterns on EEG look like at this stage. Other than we've just been using this to look back in terms of the timing of isoelectricity as compared um, to systemic hemodynamics and brain hemodynamics. So in terms of more granular things, I don't have anything fancy to show you or great insights, unfortunately. So yeah, but I think this is an invaluable sort of pairing of electrical and circulatory data that we'll have to get into. We've only done the depth electrodes on 3 cases so far. Gwendolyn asked, did brain oxygen extraction vary over time? So as a cohort, there appeared to be less brain O2EF changes with hypotension compared to systemic. Again, with the caveat that the brains of these patients are severely injured. So that plus the sedative use likely, I think, explains this finding. Now, this is just the summary data across the cohort of 16. However, in the paper, we have the individual data as an extended panel, and there is quite a bit of individual variation. So that was kind of the theme of this, that the data doesn't really fit with one nice pattern. This is all very exploratory, but I mean that's just the cohort, and that's just the study in science, I suppose. But yeah, by and large, brain O2F was less than systemic O2F, which I don't think was that surprising. Thanks, Mix. Hi, Erta. I missed the initial part. Explain how long after rest do you start TCD monitoring? Oh, so no, we start TCD monitoring for about half an hour before palliation, and then continue it after a rest for at least 5 minutes. So this is obviously not post-ROS because we're not ethically, we can't resuscitate these patients. So it starts half an hour before, and then at least 5 minutes. If not, we try longer to keep it on after total circulatory rest and asystole.
Cool. So we look always at and we're always amazed at the complexity of those pictures of the room. And there's, yours, I think, are the most complex that I've ever seen. It's just unbelievable. But then, we're data geeks on our side. And so I always wonder, okay, well, if it's that complicated to collect all the data out the back end, there's got to be just tremendous complexity in gathering and harmonizing. Can you describe a little bit how you approached that side of it?
Yeah, I mean, this is the side of it that we struggle on, right? Like we have like 4 different computers collecting data and pulling data out of different monitors and trying to time align it, right? So like, I mean, we do our best. It's pretty rudimentary. So this is why I was really excited to present to you guys, because I mean, my goodness, if there's people who've figured out how to harmonize different data stream collections from various different sources, it's yourselves. So the hemodynamic data, telemetry data is collected on ICM plus, the TCDs separate, the SJVO2 and the pulmonary artery catheters are on 2 separate Edwards hemispheres. So we pull the data from different monitors and then try to time align it. one of the problems with that is, of course, the granularity is different from all these different monitors. So for this initial cohort, for example, the SJVO2 and the cardiac output data is averaged over every 20 seconds. We don't have it more granular than that. So that's a problem. Same thing with the NEARS data. So could there be more interesting relationships there that we're missing because of our lack of granularity? Of course, absolutely. But this initial sort of cohort was just a proof of concept. We just wanted to see, okay, can we actually do this? And the answer is yes. And now we're at the stage of like, okay, like let's continue to do this, but really um, uh, take our data output strategy to the next level. And that's where I'd be super keen to collaborate with yourselves.
Well, we would be too.
Hey, Mip, this is Dick.
Dick.
Nice to talk to you again, and thanks so much for this presentation. Really outstanding. You know, one of the things I think we've talked about is, you alluded to it in your presentation about, you know, kind of the troponin for the brain, or, you know, some kind of indication of brain death that's maybe more, more accurate. One of our interests, as you know, is outside of the hospital, like in austere care. You could, you know, envision Ukraine, where somebody gets a head injury and they don't get to the hospital. They have to manage them in a pretty rudimentary setting.
Yes.
And you know, GFAP, you can't, that's not practical yet out in the battlefield, although they do have a handheld unit for that from Abbott. But do you think that through your work, there would be, you could distill down more of a physiological biomarker that they could use for triage?
Yes.
So maybe comment on that.
Yeah, absolutely, Dick. Like, I think this is where, like, the power of these, like, I've viewed this this sort of study as an opportunity. Like, it's almost impossible to, I mean, it's not ethically possible to make the healthy human brain ischemic and then say, okay, we're gonna collect serial blood samples on people and see what the brain's spitting out, right? Like, so whereas this is like the decision for palliation has already happened, and what we're doing here is just asking for the opportunity to study in a comprehensive manner. Now, the proteome doesn't seem to change. So two caveats. The proteome doesn't seem to change, but I think that this is just like, these patients some are passing away very quickly, some are passing away over 2 to 3 hours, some longer. I think in the ones who are passing away very quickly, it's just not enough time for transcription, translation, and then efflux of the new proteome signatures to occur. And perhaps the blood-brain barrier is intact. Varying degrees of disruption are intact in some patients. However, I think this is why I'm really excited about the metabolomics work we're going to do this fall on these same samples, because if something's going to change, the metabolome should change quicker. And perhaps there could be, I suspect there will be a lot of changes in there. And the reason I say that is because even on our microdialysis data, we see changes in brain lactate, we see changes in brain glucose and pyruvate that occur during this process. So I suspect the metabolome will change, and there could be a biomarker in there that would be useful. One of my I don't want to call it a dream, but one of my goals has always been to provide every day in ICU, like, we round, and even earlier today, and we have like liver panels and creatinine and kidney panels and blood tests for everything. We don't have a blood test for the brain in that moment necessarily, and so this, I think, can potentially provide, as close to possible, a human model to identify either protein or metabolite that could be used for that triage, as you allude to. And like, what I think about, like, this is one of the important things for our jurisdiction. Like in British Columbia, we have like densely populated areas with rural areas and that are hard to get into with like medevac flights. And I used to do air ambulance, and you get calls all the time, be like, "Oh, I think they're having a stroke. I'm not really sure. We don't have a CT scan." Like, my goodness, wouldn't it be nice to have a non-invasive blood test metric to risk stratify those patients, or on the battlefield, or in other areas where the care is maybe not as easily accessible.
Yeah, that makes sense. Thanks so much.
Okay, well, Mip, thank you so much. It's just fascinating work. And it's clear how much care you have for these patients and their families, and it's just so impressive to us to see, like Jamie said, that they become your families. So anyway, I just wanted to
Thanks.
say that. Yeah.
Yeah, if I may, just one last thing is, like, this is, look, our research team doesn't have some sort of fascination with this kind of physiology. It's we conduct this as much as possible in a very patient-centric manner. And I'm glad you brought up the families, because we do stay in touch with them. We do connect with them, and conduct follow-up interviews in terms of how this process was and participating in the study was for them. So, in many cases, they've ended up being additional families have ended up being patient partners to advise this work. So we try to do it in as much of a thoughtful, respectful, and patient-centered way as possible.
Well, it's clear you're succeeding, and it takes a certain type of bravery to go and approach this topic with families who are facing these situations, and to do it successfully. It's just amazing work. Aside from the science, there's just a huge amount of compassion and humanity that goes into it, and you clearly are doing that very well. So thanks very much.
Yeah, thank you, guys, and yeah, would love to work with yourselves.
Cool. Are you going to be at NCS?
No, unfortunately not. I've got to pick and choose a little bit where I go. So unfortunately, I won't be at NCS.
Okay. Well, hopefully we'll get to see you.
Yeah.
All right. Thanks very much again, and we'll end it here. And everyone have a great rest of your Friday and a great weekend.
Thanks, everyone.
Thanks.